Have you ever really stopped to think about how the information packed inside your DNA, this tiny microscopic double helix, actually builds you? I mean, everything from your hair color to how your body fights off a cold, it all comes down to a fundamental process. Today, we're going to unlock life's ultimate instruction manual, a concept known as the central dogma of molecular biology. So let's put it this way. How does a simple chemical code, just a long sequence of letters, turn into a physical trait that you can literally see in the mirror every single day? Well, the answer to that question lies right at the heart of what makes us who we are, in a concept that maps out the flow of all biological information. Okay, so the key to understanding all this really started to come together back in the 1950s. This was right after the structure of DNA was discovered, and suddenly, scientists had a clear, almost elegant framework for how life works at the tiniest, most fundamental level. And the person who proposed that framework was none other than Francis Crick, one of the guys who discovered DNA's double helix. In 1958, he came up with the term central dogma. He basically saw it as this fundamental one-way street for genetic information. It was the first real roadmap from code to reality. So what did this original blueprint actually look like? Well, it was beautiful in its simplicity, and for quite a while, it seemed to explain pretty much everything perfectly. Crick's original idea was a strict one-way flow. It goes like this. Information is permanently stored in DNA. Then it's copied into a temporary, disposable message called RNA. And finally, that message is used by the cell's machinery to build a protein. And proteins are the tiny machines that do, well, everything, from digesting your food to contracting your muscles. It's a straight line from storage to action. It all starts right here with DNA. The best way to think about it is as the master blueprint, or maybe the permanent hard drive of the cell. All of its information is written in a 4-letter alphabet, A, G, C, and T. The unique sequence of these letters contains the instructions for building every single part of an organism. It's that simple and that complex. Now, you wouldn't take the master blueprint to a messy construction site, right? It could get damaged. So the cell does something smart. It makes a copy. This first step is called transcription. Imagine photocopying a single page from a giant encyclopedia. The language is the same. It's still written in nucleic acids, but now you have a working copy, a messenger, that you can take out and use. So next up, that mRNA message travels out to a molecular factory called a ribosome. And this is where the magic happens. This is translation. Here, the language of nucleic acids gets converted into the language of proteins, a specific chain of amino acids. This is the moment, the crucial step, where the abstract code becomes a physical, functional thing that can actually do a job in the cell. You know, this analogy just works so well. Think about it. Your DNA is like that final essay saved on your computer's hard drive. That's the permanent storage. When you hit print, your computer sends a message to the printer. That's your mRNA, a temporary instruction. The printer itself, that's the ribosome, the machine doing the work. And the final printed out essay? Well, that's your protein, the tangible, functional product that actually does something. For years, this simple, straight-line model was the absolute bedrock of biology. But, science is all about pushing boundaries and questioning assumptions. And pretty soon, researchers found some fascinating exceptions, some major plot twists that made this whole story a lot more interesting. And this really shows you the difference right here. That original one-way arrow from DNA to RNA suddenly got a new arrow pointing back the other way. It became a two-way street. Scientists discovered that, in some cases, information could flow backward, from RNA back into DNA. And not just that, RNA could also make copies of itself. This simple line was quickly becoming a complex network. The first huge challenge to the dogma came from a pretty unexpected place: the study of these really strange viruses called retroviruses. And the most famous one of all is HIV, the virus that causes AIDS. See, these viruses carry their genetic code as RNA, not DNA. So to take over a host cell, they have to somehow get their code into the host's DNA blueprint. And they do this using a really special enzyme that performs reverse transcription. It literally reads the viral RNA and writes it backward into DNA. This was a complete and total violation of Crick's original one-way rule. And the thing is, this wasn't just some minor detail.
The discovery of this enzyme, which they called reverse transcriptase, by Howard Temin and David Baltimore, well, it was so revolutionary it won them a Nobel Prize. It proved without a doubt that the flow of information was not a strict one-way street, and it forced the entire scientific community to basically rethink the rules of life.
But the surprises did not stop there. It turns out that humble RNA molecule had another big secret. We all thought it was just a passive courier, just carrying messages from point A to point B. Turns out, it can also be an active worker. Scientists discovered what are called ribozymes. These are RNA molecules that can act like enzymes, speeding up chemical reactions. Some of them can even cut and splice themselves, editing their own messages before they get translated. So RNA wasn't just a piece of tape carrying a message, it was a pair of scissors too. And again, this was a huge deal. The discovery of this catalytic RNA by Thomas Seck and Sidney Altman also won a Nobel Prize. It revealed a whole new layer of complexity to the system. RNA wasn't just a messenger, it was an active participant, a tool and a message, all rolled into one. So you might be thinking, okay, these are just some nerdy academic details, right? Exceptions to the rule. But understanding these twists in the dogma has had massive real-world consequences. It's literally transformed medicine. Just look at this. Understanding reverse transcription is how we fight HIV. Drugs like AZT are designed specifically to block that one enzyme. Those RT-PCR tests that we all became so familiar with recently, they use reverse transcription to find tiny amounts of viral RNA. And now, scientists are engineering ribozymes to act like molecular weapons, targeting and destroying the RNA from viruses or faulty genes. It's amazing. So where does all this leave the central dogma? Is it a broken law? Not at all. Actually, it's a perfect example of how science is supposed to work. Even with all these new discoveries and modifications, the central dogma is still a foundational concept. The main pathway, DNA to RNA to protein, is still the primary way information flows in pretty much all life on Earth. The exceptions didn't break the rule, they just made the map more detailed and way more accurate. And that's such a cool thing to remember about science. Its principles aren't rigid, unbreakable laws. They're models. They're our best current explanation for how the world works. And as we discover more, we don't just throw the old map away. We refine it. We add new roads, we explore new territories, and we make it better. And really, that's the thought I want to leave you with. We started with a simple, straight line. Now, we see a complex, interconnected network of information flowing through our cells. It just makes you wonder, right? What other pathways are still hidden, waiting for us to discover them in the incredible machinery of life?